Mendel's work with the garden pea and the laws of segregation, the laws of independent assortment that he discovered, really helped establish the field of genetics and helped us understand some basic principles about genetics. But there's a lot of limitations to what Mendel did. First of all, it turns out that he always studied traits that differed with regard to only one gene. So he was looking at traits that were only controlled by one gene. And that greatly simplified his studies and his results. But now we know that most traits are probably influenced by many genes. So 2, 3, 4, 5, 6 dozens of genes might control traits. And so most of the things that we inherit are not as simple as what Mendel studied. So they're not going to follow Mendel's laws. So we call this non-Mendelian inheritance. The first couple of examples we're going to look at are examples of gene interaction. These are traits that are controlled by 2 or more genes, each of which has at least 2 alleles. So the 2 examples of gene interaction that we're going to look at, so we have gene interaction as a broad category of non-Mendelian inheritance, and the 2 examples are epistasis and polygenic inheritance. So the first one is epistasis. Epistasis is when the alleles of one gene or the outcome of one gene masks the outcome of another gene. And this is usually because there's 2 different genes that contribute to an outcome. So in this case, where it was first discovered was in sweet peas. So I know this is confusing, but this is a plant that's related to garden peas, but not the same, called sweet peas. And the researchers took 2 white flowers and that were true breeders. And if this was following Mendel's laws, if you took 2 white flowers and crossed them, you would expect to see a white flower. But instead what they saw was a purple flower. So that was the first confusion. They had 2 whites and suddenly saw purple. And then if they self-fertilized the purple, they also did not see a 3-to-1 ratio like they would have expected. Say they saw this very complicated ratio of purple to white. And what they realized is that there's 2 genes involved in flower color in sweet peas. There's the C gene and the P gene. And I kind of like to think of this as 2 questions. The C gene controls whether or not there will be color. So C for color, yes or no. Only if the answer to the C gene is yes, will the P gene matter, because the P gene is then purple or white. So the C gene comes first, and that controls whether there will be color or not. If the C gene says yes, then the P gene will decide whether that's purple or white. If the C gene says no, then there'll be no color. So therefore, it will be white no matter what the P gene says, because in nature, white is really the absence of color. So let me show you how this works. So making flower pigment is really a 2-step process. So this will make purple pigment. The first step of it is controlled by the C protein, which is made by the C gene. And the second part is controlled by the P gene that makes the P protein. So if I make C enzyme, so if the answer is yes to that question, like I was saying, the 2 questions, then the next step will be the second step, and then this will decide will there be purple or no color. So purple, if this gets made, so it will make purple pigment, or this will not make the P gene and then it'll be white. So there's 2 ways to get white basically. Or rather, you have to have this and this saying purple in order to get purple. If the C enzyme or the C gene says no, then I don't make this. If I don't make this, I never get to the next step. So basically, you can't get to the second step if you can't get past the first step, and so I'll never make purple pigment if the answer to this question is no, even if this one says purple. So what that means when we're interpreting genotypes, so let me write out some genotypes here. Oh, I already did that one. So looking at genotypes, the C gene says yes, so this is a yes to color, and the P gene then says purple, P for dominant. So this would be a purple flower. So if I look at this one, this says no to color. Even though this gene says purple, because this says no, this flower will be white. So again, I don't get past this step, so what this one says doesn't matter. So this flower will be white. If I look at this one, again, this says no. So even though this says purple, it doesn't matter. This one will be white because I can't get past this first step. This one will also be white because it's both no and white. And this one will finally be purple. So it says yes to color. And then it says purple. So this will be purple. Let me give you one last example here. What color will this one be? It says yes to color, but then this part says white. So this will be a white flower. So basically, again, the expression of one gene masks the expression of another. So if I look at this one saying white masks this one saying purple. So that's why it's epistasis. This is the mechanism of Albino Ism, or being an albino, in animals and humans. So, for example, somebody's background skin color might be to be very dark-skinned, but if they have the albino gene, that's upstream from that, and so their whole body will be very light-colored, even though they have dark-colored genes. And there's a picture in your textbook of this, an example of this, a little girl with her sister. She's African background, so her genetics is for dark skin, but because she has albinoism, that's upstream, and so it masks all the other genes for coloration. Same thing is true in lots of animals that also have albinos, like rabbits and rats and mice. So the second example of gene interactions is polygenic inheritance and continuous variation. So there's two types of traits. What we've looked at up until now is discrete or discontinuous traits. These are where you have like distinct categories, like purple or white. You don't have anything in between, you just have purple or white flowers. You can also have, for example, in fruit flies, red or white eyes. There's nothing in between, they're either red or white. That's discrete or discontinuous traits. Many of our traits, probably the majority of our traits, are continuous or quantitative. That means that they're really a range rather than discrete phenotypes. So think about height, skin color, the number of apples on a tree, the number of flowers on a plant. These all have a range. It's not that you're exactly 6 feet and somebody else is exactly 2 feet based on their genes. There's continuous variation over the range of phenotypes. This is usually the result of a polygenic inheritance. Polygenic means many genes. So many genes control the trait. So for skin color, for example, there's at least 3 genes known to control skin color. And for simplicity, they use the A, B, and C in your textbook. So there's the A gene, the B gene, the C gene. So somebody who is homozygous dominant for all three would be the darkest. Someone who is homozygous recessive for all three is the lightest. And then you have every variation in there. So this person has one dominant, they would be slightly darker. Same thing with this person. They only have one dominant, so they'd be a little darker than this person. And same thing with this one. They've only got one dominant, so they'd be a little darker than this person, but these three would be about the same darkness. So looking at this chart from your textbook, they would be these ones. So there's the extreme lightest, and then the three genotypes that are a little darker. And then again, you can have all these different ways to get to a slightly darker shade, and then all these other ways to get to a slightly darker shade. And then working back from most dark, a little lighter, a little lighter. So everyone is somewhere in here, right? And then notice though, it's not like we're all like one step away from the same shade, like that we're all the same color. If like these people are all the same color, there's a significant influence of the environment, and that's what makes this be a smooth curve and have us all have sort of unique skin color. And then the interesting thing about skin color is the whole range shifts this direction darker in the summer. So the whole average skin color of the whole earth shifts a little bit to the right in the summertime. So again, this is continuous variation where we have a range within the traits. We're all some variation between light and dark, not just two distinct. You're either this color or this color. This continuous variation is usually the result of polygenic inheritance, when many genes control a single trait. In the case of skin color, there's 3 genes that control the trait, and we all get some combination that gives us a unique starting point for our skin color, but that the environment will then also play a huge role in.